Thank you. Good morning, everyone. It's nice to see everyone here. I'm Jenny Wood. I'm one of the faculty members in the Department of Psychiatry, and it is my pleasure to introduce Dr. Ouellette today. She's one of our amazing PGY-3 residents, and I've had the pleasure of working with her as her long-term psychotherapy supervisor for the last academic year. Dr. Ouellette comes to us from University of Auburn for undergrad, where she majored in science, had a Bachelor of Science in Biomedical Sciences, and also got a minor in psychology. She then completed medical school at the University of South Alabama College of Medicine, and she's been with us as a resident for the past 3 years. Next year, Dr. Ouellette is going to be going to do her child and adolescent psychiatry fellowship at the University of Cincinnati. And she's going to pursue her interests, which focus a lot on adolescent female mental health. She's also very astute at psychotherapy, and we've been doing a lot of DBT together, so hopefully she'll get to expand on some of her interests in fellowship as well. Personally, she's the mother to a very enthusiastic, adorable baby, Joshua. He's not a baby anymore, he's getting big. She also enjoys in her free time sketching animals, spending time in nature, and has been just a really lovely person to get to know.
Thank
you. I look forward to hearing Dr. Ouellette's talk today. Thank you so much.
Thank you. I appreciate it, Dr. Wood. So like I said, you all know me, I'm Dr. Ouellette. My presentation is on trauma, epigenetics, and the development of psychiatric illness. As it pertains to my interests, it tends to very much connect with the adolescent population and childhood population. I'm going to talk about how the epigenetic biomarkers are actually different in children who experience trauma compared with adults. But we will get into that in a little bit. So first, no disclosures or conflicts of interest, nothing to disclose. Objectives. First, I'm going to define traumatic events that can precipitate the development of PTSD, as well as its epidemiology. I'm going to discuss an overview of the diagnosis, cardinal features, pathophysiology, and comorbidities, and we'll go deeper into the pathophysiology later on. I'm going to discuss various treatment options for PTSD and current research into new options. And then we're going to get into the epigenetics, how these modifications are implicated in the maladaptive reactions to stress, go into the biochemical processes involved in gene translation and epigenetic modifications. And then we'll discuss how this affects the hypothalamic pituitary adrenal axis, immune regulation, and fear conditioning, and how this all relates to heritability through epigenetics, how this can confound among generations from parent to child, and then discuss how this research could potentially lead to more individualized prevention and treatment for vulnerable individuals. So first, just a basic introduction. You all know what PTSD is: a stress-related psychiatric disorder thought to emerge from a complex interactions among traumatic events and multiple genetic factors. It's the classic nature versus nurture debate, but an increasing body of evidence supports that the role of epigenetic regulation, which is basically nature and nurture. It's how does your nurture affect your nature, and then how does that further affect your nurture? So it really kind of combines the picture. And then we think, disentangling those mechanisms could potentially enable the identification of individuals who would be most at risk for maladaptive responses to trauma. And if we were able to understand enough of it, potentially guide the direction of new treatments. So basic overview of trauma as defined for PTSD, because trust saying trauma can be a little vague. It's an event that can lead to PTSD is defined as an intense threat for their own life, health, or physical integrity, and goes along with feelings of strong fear and helplessness. And also immediately witnessing such an event can lead to traumatization in the absence of personal victimization. This wouldn't include like watching it on the news. And then PTSD has been described as the complex somatic, cognitive, and affective behavioral effects of psychological trauma. It's characterized by intrusive thoughts, nightmares, flashbacks, and avoidance of reminders, hypervigilance, and sleep disturbances, all of which lead to significant dysfunction. So what's notable is traumatic events, by this definition, have been estimated to occur in 90% of individuals in the general population in some point in their lives. So experiencing trauma doesn't mean someone's going to get PTSD. So then it's the question of why. What leads to developing PTSD versus what leads to, you know, somebody having resilience? Because some people undergo traumas and grow from it and actually become more resilient to future traumas. The 1-year prevalence of PTSD is about 3.5 to 4.7%, while the lifetime prevalence is about 6.1 to 9.2%. And then I've got a chart over there that talks about, you know, different percentages of people who tend to be most affected by it. And notably, you know, children and battered women are at the highest, also victims of sexual assault. Something else of note that I was reading about is that that victims of natural disasters, despite, you know, having events that can potentially, you know, be horrific, often have lower rates of PTSD, which they think that is attributed by the amount of social support and resources that go into it as a mitigating factor. So I think one of the biggest factors is the sense of helplessness, which makes sense with children and, you know, victimized women. Okay, so these high trauma rates together with the relatively small prevalence of PTSD indicates that trauma exposure does not necessarily lead to PTSD, like I was talking about before. As generally observed in psychiatric disorders, trauma exposure has a stronger impact in more vulnerable individuals with a higher probability to develop a pathological phenotype. The same way that, you know, or in a similar vein that somebody who, you know, has developed obesity, you know, their genetic propensity for diabetes, you know, contributes a lot there as well. And then that ability to predict individual responses to traumatic events, which are ubiquitous and often inevitable, could offer the opportunity to target preventative strategies and early interventions to vulnerable individuals. So again, here's you know, with PTSD, some, you know, with people who are afflicted, here are the most often causes, and again, sexual violence, interpersonal violence tend to be very high. So, which again, I think is related to that sense of helplessness. So risk factors for developing PTSD include a history of prior trauma, less education, lower socioeconomic status, childhood adversity, personal and family psychiatric history, female gender, African American race, poor social support, physical injury, and the initial severity of the traumatic event. Here's the DSM criteria. Exposure to the traumatic stressor that it has a threat of death, like I talked about, or actual death, threatened death, serious injury. They have to have recurrent, unwanted, intrusive thoughts, flashbacks, traumatic nightmares, avoidance behaviors to avoid stimuli related to the event, cognitive distortions about not being able to recall certain features, and or negative beliefs about the world. Increased arousal leading to irritable and aggressive behavior. And then the duration has to be present for more than 1 month. Otherwise, it's acute stress disorder, which often resolves. And then it has to lead to some sort of functional impairment and not be caused by another condition. So with this chart, did they have a traumatic event and were they exposed to it? Do they meet the symptom criteria? Has it been more than a month? And is it causing impairment in their life? Which the impairment, you know, is important as with regards to any mental illness, and that's, you know, for leading to the diagnosis. So here's the cardinal features, like I discussed in the diagnosis: intrusion symptoms, avoidance symptoms, negative alterations in cognition or mood, and arousal or reactivity changes. And then, you know, complex PTSD refers to the proposed subtype with prolonged traumatization, often during developmental periods. Additional symptoms include dissociation, emotional dysregulation, somatization, altered relationships, and altered systems of meaning, which if you look at the epigenetic mechanisms, which I go into later, there's actually, you know, distinct pathways related to childhood prolonged trauma versus the trauma of veterans and PTSD. Pathophys, while most of the pathophysiology is unclear, initial research findings are accruing. MRI studies showed decreased volume of the hippocampus, left amygdala, and anterior cingulate cortex, which potentially relates to poor memory with the trauma with the hippocampus, emotional dysregulation with the amygdala, and an overactive, quick behavioral response and poor judgment in the anterior cingulate cortex. There's also been shown to have hemispheric lateralization with the left hemisphere, which could relate to the problems with perceiving time. And then we'll get much more into the pathophysiology of this later, but there is noted to be an increased central norepinephrine levels with downgraded or downregulated central adrenergic receptors, and chronically decreased glucocorticoid levels with upregulation of their receptors. And of note, there is a anecdotal finding that autoimmune disease tends to be increased in this population, which I suspect is related to the dysregulation of the HPA axis in the immune system. Comorbidities, 16% of these patients have one coexisting psychiatric disorder, 17% have two, 50% have three or more. Depressive anxiety and substance are 2 to 4 times more prevalent. Personality disorders are greatly, increased prevalence. And somatic symptoms are reported to be as much as 90 times more likely, which is, I believe, really notable. Course and complications, usually a chronic condition, only about 1/3 of patients recover at 1-year follow-up, and 1/3 are still symptomatic 10 years later. And then most experience it quickly, but there is a delayed subtype that has been noted. And it can have a tremendous economic burden, with the loss of productivity, the healthcare costs, the unemployment, the caregiving. And so it can be a massive problem for society. Potential treatments, we'll go through these kind of quickly. So first line is typically SSRI or SNRI. It has the most data. It can help mitigate some of those negative responses. There hasn't been much comparing the two. Another first line is trauma-focused therapy, though often you'll prefer medication in addition to it if they have comorbid psychiatric symptoms, which they often do. Insufficient evidence of other antidepressants besides just the SSRIs and SSRIs, but again, useful for treating comorbidities depending on the patient. So trauma-focused therapies include cognitive behavioral therapy, exposure-based therapy, and EMDR. There's high evidence or strong evidence supporting that these are helpful, but not really strong evidence to support one over the other. Then prazosin, often used to augment the SRIs and individuals with PTSD who had experienced significant sleep disturbance, tends to work notably about 50% of patients, at least reportedly. I've noticed that the clinical observations tend to be better than some of the data. Like the patients say, "Oh, this really helps me," and you notice, so you give it, but the data is not quite as strong as what they say. But it seems to work out. So. And then, of course, you have to worry about blood pressure when using this medication for if they have hypotension or about rebound hypertension. Antipsychotics. In individuals with PTSD or prominent psychotic symptoms, you can augment the SRI with an SGA. There's, been a lot of data coming out lately about that augmentation like they do for depression. And, the sertraline brexpiprazole combination pill is currently under review by the FDA. So current research. A lot of the, data for ketamine and the psychedelic assisted therapy, it looks pretty good, but there's the issues with blinding and with placebo effect. And then there's also the issues with like why the MDMA got rejected, because there's concerns for potential for abuse and people are kind of wary of that stuff. So I think there's a lot of potential to benefit, but I think there's also a lot of barriers to the treatment. So and then there's also current research into using propranolol with memory activation to try to tone down the noradrenergic drive. I think that could potentially be really cool. And then TMS has shown mixed results. It shows that there's a lot of improvement with the treatment, but also from the sham treatment. So I wonder how much of that is treating comorbid depression or just the behavioral activation of getting out every day and seeing somebody. So results there are kind of mixed. And then, of course, you want to avoid benzodiazepines, especially, this is a patient population that can very easily become dependent on them. And then current evidence does not support cannabis use for this. That may change in the future, but as of right now, it doesn't support the idea that it's overly helpful. Okay, so now let's get into the meat of this lecture. So epigenetics. Epigenetic changes can provide a molecular mechanism for the development of PTSD after exposure to trauma. An adaptation to a stressor, an organism can react with epigenetic changes that lead to changes in gene expression profiles and in consequence to alterations of biological pathways and neuronal functioning. And these epigenetic changes can have short-term and long-lasting effects on neuronal function, brain plasticity, and behavioral adaptations to psychological stressors. So here's just an example over there. There's maltreatment, they're exposed to a stressor. They undergo epigenetic changes, which can then change their affective and behavioral phenotypes. These are some genes that have been noted, like studies have shown they have been implicated in systems that are involved. We will talk about a few of those, but not all of those later, because the data is quite extensive. So, but also still in the early stages. First, let's do a quick cell biology review. everyone remembers this, but a gene is a segment of DNA, provides instruction for protein synthesis, consists of 4 bases: guanine, cytosine, adenine, and thymine. They're repeated in each sequence. Cytosine methylation is of particularly importance with epigenetics. And then there's the protein synthesis goes from transcription of DNA to RNA, and then translation of RNA to a protein. And then in the first step, transcription factors within the cell read and interpret the DNA instructions, recruit RNA polymerases, which facilitate the copying of DNA into mRNA. So little cell biology review. And those can be, proteins can be modified post-translation. And here's the review of epigenetics, of the basics of it. So the main functions of the epigenome are to regulate gene transcription and to compact the DNA into the cell nucleus. The main processes involved in epigenetics are DNA methylation and hydroxymethylation, histone modifications, ATP-dependent chromatin remodeling, and then the use of non-coding RNAs. These processes are shown to be influenced by environmental exposure and collectively shape the transcriptional activity of genes without changing the underlying genetic code. And what's most interesting is, there have been studies that have shown that these modifications can, in some cases, be passed on genetically. So if a mother had a exposure to a traumatic event and it led to increased methylation of her DNA, that pattern of the DNA could be passed on to the child. Okay, so here's just a basic little diagram of histones and DNA and how the gene is active when the DNA is accessible, but when the chromatin is packed together, then the DNA is not accessible and unable to be transcribed into proteins. So just do a quick overview of the basic parts. DNA methylation is the one that has had the most studies with epigenetics and PTSD. It's the covalent modification of the cytosine residues, and it's mediated by the DNMT3A and DNMT3B. They add a methyl group to unmethylated is usually associated with repressed gene expression, can interfere with the binding of transcriptional enhancer complexes, which means that increased methylation means that the gene is not transcribed as much. It's blocked off. Histone modification. Histones are like the beads on the string, or they form nucleosomes that are the beads on the string, and chemical alteration of those through acetylation or deacetylation influences the structure of chromatin, which allows there to be access to the gene or not. Generally, acetylation opens up the gene. Deacetylation kind of closes it off. And then there's also histone methylation, or methylation, which is thought to have an opposite effect to acetylation and also kind of closes it off. So it's a non-coding RNAs are transcribed from DNA, but unlike the RNAs involved in protein formation, they are not translated into polypeptides. They are functional or involved in the regulation of other RNAs, like mRNA, tRNA, and rRNA. So overall, the epigenetic regulation in PTSD has been supported mostly for genes involved in stress responses, neurotransmitter activity, immune regulation, or repetitive genomic elements. Like I said, most of them have been involved in methylation. There's quite an extensive, body of research out there on various methylation patterns. And then histone regulation has also been implicated in some of them, and so has involvement with the non-coding RNAs, as I've written up there. But it's mostly DNA methylation, so that's what we're going to focus on. The hypothalamic pituitary adrenal axis, that is one of the main systems activated after exposure to a trauma. And this system being disturbed has frequently been described in PTSD. It has particularly been associated with suppressed cortisol levels that have been attributed to hypersensitivity of the glucocorticoid receptor and enhanced negative feedback inhibition. These HPA axis abnormalities may drive persistent changes in the neuroendocrine milieu, which could affect epigenetic changes in cells throughout the body. So in a way, this is almost like akin to tardive dyskinesia. I mean, also different, but the hypersensitivity of the glucocorticoid receptor is almost like those abnormal movements that developed with tardive dyskinesia. So that has that overreaction to something, small. When there's suppressed levels, they'll still have that reaction. Okay, so I'll go through this quickly. Hypothalamus is the superior center of the axis. It produces the hormones, corticotropin releasing hormone and arginine AVP. They act together in the stress reaction to stimulate the distribution of ACTH from the pituitary gland. ACTH triggers the release of sexual hormones, aldosterone and cortisol from the adrenal glands. So it goes from the hypothalamus to the pituitary to the adrenal glands. And then the cortisol acts with the catecholamines, which are involved in the noradrenergic response as primary effectors of the stress system. And they bind to the glucocorticoid receptor and the mineralocorticoid receptor and act as transcription factors. And this binding allows them to regulate adaptive responses to stress, including metabolism, immune activation, and cell proliferation. And then there is a negative feedback system via the neuronal glucocorticoid receptors. And so generally a decrease in their expression is associated with the response to stress due to an impaired negative feedback. So here's a chart. Starts in the hypothalamus, CRH goes to the anterior pituitary gland, ACTH goes to the adrenal glands, release glucocorticoid receptors and mineralocorticoid receptors, and this leads to effects throughout the body, which then regains homeostasis through a negative feedback system. So let's talk about some of the data on epigenetics. So among genes involved in the regulation of the HPA axis, one of the most extensively studied is the gene encoding the glucocorticoid receptor NR3C1, which is regulated through multiple promoter regions located in its non-coding exon 1 variants that contain numerous glucocorticoid receptor elements. Particular attention was drawn to this locus by studies in rodents and humans linking early life adversity with increased methylation levels of the NR3C1 exon promoter in peripheral blood and brain tissue. And a study examining combat veterans, subjects with PTSD were shown to have lower peripheral blood methylation levels and compared with combat veterans without PTSD. And the methylation inversely correlated with the dexamethasone suppression of cortisol and PTSD symptoms. In another study, an increase in the methylation levels predicted favorable PTSD response to prolonged exposure therapy, supporting it as a biomarker. So some of the data is a little mixed, like some, it'll be higher when they've had the traumatic exposure, but then it also doesn't necessarily get lower. It might get higher again in after therapy. So it's useful as a biomarker, but not always as, it's not always super clear cut. And in line with these findings, civilians with a lifetime diagnosis of PTSD were shown to have lower T cell exon methylation levels and higher glucocorticoid receptor expressions. So the gist of this is that, the glucocorticoid receptor is very heavily implicated in the stress response with the HPA axis, and epigenetics have been shown to be involved in affecting the glucocorticoid receptor in response to a stressor. So another glucocorticoid responsive gene that has drawn increasing attention is the gene encoding FK506 binding protein 51. It's induced by glucocorticoid receptor activation and acts as a co-chaperone for it, and is involved in the negative feedback loop that decreases glucocorticoid receptor signaling and maintains homeostasis of the HPA axis. And epigenetic changes with this could be involved in dysregulation of that HPA axis. So supported in many studies showing that the alleles confer increased risk for PTSD, especially in the context of early adversity, and are associated with worse PTSD symptoms. CPG sites located near glucocorticoid response elements were shown to undergo demethylation in response to childhood, but not adulthood trauma in risk allele carriers, which I found really interesting, that there was a distinct difference between patients who experienced the trauma in childhood and patients who experience the trauma in adulthood, which makes me wonder if that might be part of the difference between the PTSD versus the complex PTSD picture. And I think that that's something that's worth, could be potential for a lot of research later on. Similar DNA methylation changes are observed after glucocorticoid exposure in human progenitor cells, suggesting that the demethylation in response to excess glucocorticoids following early life trauma could be an epigenetic signature that's stable across tissues. And then levels of the promoter were shown to accompany treatment response, suggesting, again, potential usefulness of this marker in tracking disease course. Though a lot of these studies have not been heavily powered, which is a lot of the weaknesses of these studies is they haven't been heavily powered, but there's a lot of potential for more new studies like genome-wide studies in the future to be more powered. So rodent studies in particular have provided valuable insights into the neural circuitry and epigenetic mechanisms that are involved, especially in the fear response. As you can see, these studies are kind of sad, but they train rats and have used those as a model for PTSD and fear conditioning. They put them in a box and then they train them to be scared about being shocked. And it's pretty sad, but but it has been very helpful for PTSD research. And one study found that DNA from peripheral blood, higher methylation levels within the promoter region of catechol-O-methyltransferase, the enzyme responsible for the inactivation of the catecholamine neurotransmitters, are associated with impaired fear inhibition, which makes sense that if you're not able to inactivate the adrenergic response, that it would impair your fear inhibition. So there's definitely an aspect of fear conditioning in PTSD, which is what, some of the therapies, would be most effective at tackling as compared to medications. The learning of the fear, having it reinforced over and over and over versus if it's a one-time thing and you can kind of have extinction of that response. Okay. It's also been examined at the level of ADCYAP1, pituitary adenylate cyclase activating polypeptide, which is a gene that induces activity in the pituitary gland, modulates stress responses, and involved in regulation of certain paracrine and endocrine cells. It was noted to be, the levels were associated with PTSD diagnosis and symptoms in highly traumatized female subjects, and also just PTSD symptoms overall in both sexes. And these were corroborated by experiments with rodents. Okay, epigenetic changes in immune-related genes have been observed in numerous studies as well. Methylation levels of the genes encoding interleukin-8 were found to increase after deployment in military service members who developed PTSD, but to decrease in deployed subjects who did not develop PTSD, which decreasing makes me wonder, is that related to the post-traumatic growth? Another epigenome-wide study showed that subjects with PTSD have differential methylation levels at other genes involved in the inflammatory process. TPR, CLEC9A, APC5, ANXA2, and TLR8, accompanied by increases levels of IL-4, IL-2, and tumor necrosis factor A. And then another one did a genome-wide study methylation profile of over 1,400 genes, and the number of uniquely methylated genes were significantly higher in PTSD-affected patients, and there was a distinct signature reflecting immune activation. So these lasting epigenetic signatures at immune-related loci might contribute to immune dysregulation and increased risk for PTSD through the HPA axis, and may also the immune dysregulation could likewise contribute to vulnerability of the development of PTSD by altering functions in the brain. So it kind of just all plays into each other. There's been studies that have shown like a kindling hypothesis that once you've been exposed to one trauma and you've haven't recovered from it, that somebody who's been traumatized by that is more sensitive to being re-traumatized, reinforcing that behavior. And then there has been data showing that people are who have PTSD and these type of situations are more susceptible to somatic conditions like cardiovascular disease, metabolic syndrome, autoimmune diseases, which could theoretically be related to these disturbances in the HPA axis. So there's a lot more research that needs to be done, but I really think that there's a lot, to be learned from this. The generational transmission and early exposure. Okay. So contrary to what may seem intuitive of a disorder that is reliant on environmental influence through a traumatic event, research suggests that genes do play a role, perhaps a significant one, in the risk of developing PTSD. In one twin study, heritable influences accounted for 46% of the variance, and one showed 71% of the variance in females. And another study suggested that exposure to assaultive trauma may not be entirely random and is influenced by individual and familiar risk factors. I kind of suspect that some of that would be related to, would be related to more your situation, but you know, there's also a temperament involved there, like risk-taking behavior. It is known that parental post-traumatic stress can cause negative psychological outcomes and potential biological alterations in their offspring, with several studies indicating that the severity of a patient's PTSD symptoms may contribute to the child's psychological difficulties. This is a bit of a nature versus nurture debate. How much of that is genetic? How much is that is the parents, generational trauma, their own trauma, they kind of project on the child, and was more of a behavioral response. But I think it's a mix of, but definitely a mix of both. But there is some data, like with Holocaust survivor victims, that there is definitely a genetic component to it that's separate from the behavioral component. So epigenetics during gestation. There have been studies that have shown that trauma exposure early in life appears to be a sensitive period that is associated with lasting epigenetic changes and more robust effects on PTSD phenotypes than trauma exposure later in life. The growing body of evidence suggests that there is innate endogenous processes sculpting the DNA methylation pattern during gestation in response to the external environment. So if mom is pregnant, mom is getting really scared, and mom is getting really stressed, then there might be those DNA methylation patterns in the baby, even though they, obviously don't remember or know what's going on. Researchers have identified DNA methylation alterations in brain tissues, including the glucocorticoid receptor, like we've been talking about, vasopressin and BDNF in animals, as well as the glucocorticoid receptor and rRNA in humans. And in these studies, early life stress and DNA methylation alterations were associated with various behavioral abnormalities in adulthood, such as increased reactivity to stress and suicide. This could potentially play a role in the pathogenesis of personality disorders, if I would suspect. Epigenetics during infancy. Rodent studies examining parental care influences revealed that differences in maternal phenotypes, namely grooming behavior, had effects on their pups' development of behavioral and HPA responses to stress in adults. Further studies examining DNA methylation in high versus low maternal care parental behaviors revealed elevated DNA methylation levels in the offspring with low maternal care and lower in those with high maternal care, which could potentially contribute to reduced transcriptional activation of the glucocorticoid receptor in low maternal care offspring. This, connects pretty strongly to, attachment theory, like, everything with, studying that about, the comfort, about the insecure attachment style versus the secure attachment style, which is mostly something that's more observed like psychologically, but having something, within like that can be physically studied, that connects to all of that, to me is just fascinating. So epigenetics during childhood, I bolded this part because I found it so interesting. One genome-wide study used a design that compared trauma-exposed control subjects without PTSD with patients with PTSD with or without childhood trauma. Patients with different traumatic histories showed distinct peripheral blood gene expression and distinct DNA methylation profiles. Compared with control subjects, patients with PTSD without childhood abuse showed 244 different transcripts, whereas those with childhood abuse showed 303. Okay. The overlap of the transcripts between the two PTSD subgroups was only 2%. I mean, to me, that's insane. Like the DNA methylation changes that accompanied these gene expression differences were 12-fold higher in the subgroup with child abuse. Everyone's kind of always known, like, childhood trauma is really bad, like the ACE study and all the concerns about it. But having like this type of data to me is just striking. A subsequently conducted pathway analysis showed several overlapping networks between the groups related to cell function and immunity. They would have different pathways, but ended up the same downstream effect. There were also several non-overlapping pathways observed, as like see below. Okay, so this evidence, alongside many other studies, show how the epigenome is highly responsive to adverse experiences during childhood, whereas environmental influences during adulthood often tend to have a less permanent effect on the epigenome and act through different mechanisms and that tend to be less stable in the development of disease. Differential biological mechanisms might be responsible for the observed gene expression differences in PTSD, depending on the time and severity of the adverse environmental exposure, and distinct biomarker profiles may be relevant to distinguish these subtypes. So this makes me wonder, talking about biomarkers, there's the worry about accessibility. Like using biomarkers to track the disease sounds really cool, and I think in the future it can be really cool. But you worry about that actually being accessible to the type of patients who would need it, since a lot of these patients tend to come from more dysfunctional places, tend to be lower socioeconomic class. So I wonder how feasible that is. But I definitely think that learning about the biomarker profiles and the systems involved in these type of responses could potentially lead to the development of new treatments that might be specifically geared towards, say, a childhood trauma phenotype or a adulthood trauma phenotype. Because our current treatments for PTSD, at least medically, they're just treating the symptoms. They're not disease modifying. So learning more about what's going on with the disease might allow us to develop treatments that are more curative. So here's an example of what I was talking about. Early trauma can affect the epigenetic mechanisms, and then whether or not you have that, you develop like risk genotypes versus protective. And then like I talked about with the kindling hypothesis, if you've already had that exposure in childhood and your body is primed to react that way due to alterations in the receptors, then the response to adult trauma could be different than somebody who had never had that childhood trauma. And I think that this could, potentially explain some of the differences in patient response to trauma and developing PTSD, and differences in the biochemical systems involved as well. So, like I said, I was talking about biomarkers. So epigenetic biomarkers, I think, they have the potential to be useful guides. I definitely think there's a lot to learn from them. And again, I worry a little about accessibility, but here's some data on them. In a study assessing epigenetic markers in veterans before and after deployment, differential changes in DNA methylation of IL-18 and H19 loci were observed in veterans exposed to trauma who developed PTSD versus those who did not. And then this is what I talked about before with the glucocorticoid receptor genes. Another study assessing them as predictors of response to prolonged exposure psychotherapy noted that pretreatment methylation levels of NR3C1 predicted PTSD responses. Changes in the FKBP5 promoter occurred concomitantly with recovery from the disorder. So tracking response, it's the type of thing, is it causation? Is it correlation? There's, a lot to be learned from it regardless. And this suggests that methylation changes can be dynamic, occur concomitantly with development or recovery from PTSD. This has implications for heritability. Like if somebody is suffering from uncontrolled PTSD, does this mean that if they undergo like therapeutic treatment, they may be less likely to pass on those epigenetic changes to their children? There's a lot, to be thought about from it. So, this can go a lot into types of prevention and treatment. If you're worried about genetic susceptibility, you could look at susceptibility biomarkers. This is kind of a future vision. And then secondary prevention, it's like somebody undergoes stress. Okay, do they have resilience biomarkers? Thanks, this chart got spelled that wrong. And then, or there's diagnostic biomarkers where okay, they probably are going to develop PTSD. And then you could get early access to treatment. And this is kind of an ideal scenario, but and then you can track the therapeutic biomarkers involved with response to see how somebody's doing. And potentially know how to treat them. That would be a really potential cool future way for this to go. Okay, so conclusion. Epigenetics provides potentially an ideal approach for understanding the interactions of genetics with environmental exposure to trauma and PTSD. The recent onset of large-scale collaborations has been supporting enormous studies in the genetic architecture of PTSD through the Psychiatric Genomics Consortium and collaborative genome-wide association studies, allowing the compilation of hundreds of thousands of samples. This would begin to allow for combined data sets examining DNA methylation arrays and RNA sequencing arrays, which soon may be very well powered. I think that, this is going to take quite a long time, but I think that the potential data from this could be very striking and could lead to a lot of new treatments in the next, in the upcoming probably decades or so. And together, such approaches offer great promise for determining the true genetic and epigenetic architecture of risk versus resilience in PTSD. And such progress may afford novel targeted therapeutic approaches to enhance treatment and prevention of PTSD in the aftermath of trauma. Thank you. How do I mute this?
Thank you, Dr. All right. Thank you, Dr. Ouellette. And we will first go with the questions here in person in the auditorium. We'll start with Dr. Belbro. And if for the virtual audience, just raise your hand and we'll get to you in just a few minutes.
Thanks, Dr. Ouellette. This was a really interesting topic, and you talked a little bit about how there have been studies showing that some of these epigenetic markers or these epigenetic changes can be reversed or they change in response to recovery. And I was curious, as you were going through the literature, did you come across studies that looked at specific treatments that we already utilize and whether those specific treatments are associated with that response, like EMDR, for example, or maybe specific like an SSRI or SNRI?
I talked about that. I think it was in in that study, it was related to trauma therapy. Where the mechanism was done with the activity
Promoter were shown undergoing. It was charted as undergoing.
Thank you for a really fascinating discussion of an interesting topic. For people that are interested in the controversies surrounding the FDA's actions on MDMA, which is really a fascinating topic, there was an in-depth article on this in the New York Times yesterday that's really worth reading. That's the first thing. The second thing is, some of you may be familiar with Siddhartha Mukherjee, who wrote a very famous book called Cancer, the Emperor of All Maladies. He also wrote a book called The Gene that he summarized in an article in the New Yorker about epigenetics that provoked probably the most degree of controversy that any New Yorker article has ever provoked, basically having to do with skepticism about this topic. That this remains a very controversial topic, especially pertaining to trauma. And there, in the scientific community, there are people that believe this is a real thing, and there are a fair number of people that think, I'm not convinced yet. So I wonder if your comments about this controversy and whether, where you see the field going with this.
I think just saying it's nothing, you know, when there is data showing these type of interactions would be also preemptive. Does that answer your question?
As much as the question can be answered.
Yeah, that's true.
Well, it's an open question. We don't
Yes.
really know where this will end up.
Mm-hmm.
And it really is fascinating. Epigenetics is clearly a topic worth research, and it is being researched,
Mm-hmm.
but it also is a controversial topic for about 1 million different reasons.
Mm-hmm.
In the current scientific climate, you know, there is concern about a return to a period of time when science is influenced by external factors.
Mm-hmm.
Political factors, for example. People call that Lysenkoism or Lamarckism.
Mm-hmm.
You know, the idea that we can influence genetics by, in essence, non-genetic means,
Mm-hmm.
or the inheritance of acquired characteristics. So it is a really interesting topic. I think there is really something to it, but I'm not sure what direction it will eventually go.
Mm-hmm. That makes sense.
All right, so yeah, very nice talk. So a couple questions. So knowing all this, you know, how much weight would you give to like GeneSight kind of study? So patient come to you saying, this is my GeneSight study, says this medicine will work for me.
Well, I see GeneSight studies, and also, like I said, with these biomarkers, I see them as potential for learning, but not like panacea, like catch-all panaceas. Like if someone comes to you and says, oh, here's my GeneSight, this is the medication that works for me, that's kind of just something that I take into account along with the rest of my data. It's like, maybe this is the case, and we could try this, and, you know, maybe, of course, GeneSight, you know, there's also, you know, the concern of is this, you know, more about your liver metabolism versus how it interacts with your neurochemistry. So I see it more as a data point. I think, you know, I don't think it's nothing. I don't think that it's everything. I think that it's a data point that you can, you know, learn from when guiding treatment. The same with like these biomarkers that I talked about. I don't think that it's something like, again, if this would be something in the future, but I don't think that the biomarkers are something you should listen to over your clinical judgment. I just think that it's something that you can use when taking, you know, information into account, kind of often how we do with a lot of lab results.
So where does all this epigenetic modification happens? You know, is it at in the parent somatic cell, or in their sex cell? You know, or it's in the, you know, like offspring? You know, so I know, why is it like, is it like long lasting, permanent? Is it modifiable? You know, sort of like.
So the answer to that is kind of everywhere, like, and it depends on the system. It's kind of in adults, it's been shown to be stable across tissues in a lot of situations. So in the somatic cells, and then that could be passed on to the germ cells. I believe it's just kind of throughout it. It is modifiable, like it has been shown to be dynamic. With showing that they were altered by, you know, the patients undergoing, you know, the psychotherapeutic treatment and having recovery from their disorders. So it's kind of a complicated, you know, tangle. It's something that, you know, it's not, you know, one specific. It's kind of interwoven with everything, with, you know, the response, and then how the body reacts and how it interacts with the endocrine system and the and everything else that's going on.
I'm always puzzled by the HPA system in trauma. Sort of like, you know, whether the cortisol goes up, goes down, you know, that kind of thing.
Notice that cortisol actually goes down because the yes, cortisol goes down, and the glucocorticoid receptors are super sensitive. Like I was kind of making the comparison with tardive dyskinesia with the super sensitive dopamine receptors.
All right, thank you.
I have a question. Actually, kind of two comments. Can you hear me?
We have a question here in the audience, and we'll get to you in just a minute.
I'm sorry. First off, thank you for your presentation. I thought you did a great job. I imagine that this topic could be kind of complicated when like discussing it with patients, if and when the research kind of does, come to a place where we are discussing this clinically with patients. Because on one hand, I feel like it could be very validating for someone who's gone through incredible trauma to realize that there is true biologic change in my body, and not only my body, but my genetic code, essentially, its expression. That's not like, something again that's all in their head or just something that happened, something bad that happened to them. But then on the other hand, that could be very scary to a lot of people and maybe feel a little hopeless, like, my genes are, just that happened. It's also about how this translates clinically and how we work with patients with, both complex and traditional PTSD. That's all. Thank you.
Thank you.
Go ahead, Dr. Bonomo.
Well, thank you for this talk. It's been very interesting. I had one comment and one question. The first was just kind of to make people think about the matter. You did a great cover of like epigenetics, and I think there is some ethical implications that we have to consider if it's true that treatment of trauma changes your epigenetics. Because the corollary of that is that if you don't treat your trauma, you pass that on to your kids, and that becomes a mechanism for karma. And so there are actually spiritual ramifications if that hypothesis is true. And I don't know whether it is. I actually think it is, but I wouldn't bet money on it. The question I had, though, was relating the HPA axis. I didn't know what you talked about with PTSD causing there to be reduced cortisol and, I guess, cortisol hypersensitivity. I wondered if you thought that was in any way related to the opposite, which is what you see in melancholia, which is where cortisol is high and they have reduced responsiveness. Do you think that there is a correlation here where they're kind of like at opposite ends of the spectrum, or is it just kind of coincidental? I just didn't know if you had any thoughts on that.
Where do I talk into? Yeah, okay. I think that could potentially make a lot of sense. It could be potentially coincidental, but considering, the super sensitivity of the glucocorticoid receptor is involved in a hyperactive stress response, while melancholia tends to have a blunted response to, most everything going on around it, and just tends to be more just kind of down, lack of energy, doesn't really care what happens to them. I think that could make sense.
Any other questions from the virtual audience? Okay, well, if there's no other questions.
There's two in the chat. Oh, okay.